from areas of high concentration to areas of low concentration. The natural movement basic premise is this. Particle theory tells us that if we have a million particles over here all bouncing around, right? And on the other side of some vessel or container or space, there are not many particles. What's going to happen over time?
Are we actually drawing this?
Sure, or don't. No, that wasn't a threat. I was just... Whoa, oh. Okay, all right. Well, I didn't know I could do that until right now. That was fun and exciting. I'm sorry. I'm sorry, I will send a sharply worded email. Dear Microsoft, it has come to my attention that you changed the picture for Class Notebook. It has further come to my attention that some people do not like it. All right, so as these bounce into each other, what's going to happen? Well, they're going to bounce off each other and then bounce off the walls and keep bouncing around. And eventually, given enough time, what should happen to those particles in this space? They're going to spread out. And not because anyone has made them spread out, not because anybody took it upon themselves to go in and move them, but just by virtue of when you're crowded in an area, you're more likely to collide. And so then when you end up moving in a direction where you don't collide with things, at that point you'll be, more stable, a low energy state. Classic example of this is grade 9 boys. Grade 9 boys, as you know, some have figured out the whole body chemistry, like body odor thing. Others are still a work in progress in that regard. But then there comes a day when some of those boys discover Axe body spray. You know about this day. Oh, you know about this day. It always seems to be the boy who has the locker closest to my classroom for some reason. And they think, well, wait a minute. If girls will like me if I wear a little bit of deodorant, they're going to love me if I wear all the deodorant. And then they spray themselves down, top to bottom, just with all the Axe body spray. And at first, where is all of that Axe body spray located? All over that little grade 9 boy, right? And then what happens over time? It spreads out. And first it spreads out seemingly into my classroom first, right? And you're like, oh, another grade 9 boy has discovered Axe body spray. Wonderful. And then given a bit more time, it will spread down to Trush's room. But of course, as it's doing that, it's occupying more volume. So the odor is becoming more diffuse, right? More spread out. And eventually, where will it end up? Throughout the whole school. And eventually, even after that, because our school is not airtight, out into the environment it will spread out, right? And somewhere there's a hole in the ozone with that little grade 9 boy's name all over it. Yeah. It's related to that, yeah. Absolutely. That's a great question. It's the same principle on which we said the phloem in plants, where if we load nutrients into one place in a closed system, they will just naturally start to spread out. And then if we actively remove them from another place, it'll naturally flow in that direction, not because we're pumping it in any particular direction, but because things naturally move from where there's a lot to where there's a little, right? This is driven by that second law of thermodynamics, entropy, the idea that the universe tends towards a low energy state. It takes energy to keep a group of molecules all piled together. And if you don't exert that energy, if you let them do their own thing, they're going to naturally spread out. So this is an example of entropy. Which is the tendency for the universe to move from a state of order to a state of disorder. Or more physically accurate, to say that it moves from a state of high energy concentration to low energy concentration. Low energy states are preferred in the universe. All right. We have a second term we're going to use during this lesson, so I want to make sure we know, which is what? Osmosis. Yeah. Yeah, and not just the diffusion of water. Osmosis is the diffusion of water across a semi-permeable membrane. We're going to do this in our second lab. Our first lab is our little testing for sugars lab that I handed out yesterday. Our second lab is going to be about osmosis. But the basic principle is this. Let's draw another box. All right. Well, actually, I don't want this to be a box because a box implies rigidity. Let's draw a cell. And let's put in the middle of that cell a membrane like this. Actually, let's just, you know what? Better idea. Let's make a membrane like this for the cell itself. And let's put that in water. That's green. I didn't want it to be green, I want it to be blue. So we've got some water here, and there's water inside the cell and outside of the cell. You see that? And we've got our cell membrane there. And then maybe inside of our cell, we've got lots of material like this. And then outside the cell, much less of it. Can you kind of picture that? Basically, if outside the cell, we have low concentration of stuff. By the way, if anyone hasn't taken chemistry, that square brackets is a way of saying concentration of. Has anyone seen that before? Did I use that in chemistry? I would have used that in chemistry. But I don't think I would have used it in grade 10 science probably. So you may not have seen that before. So we have a low concentration of stuff on the outside and a high concentration of stuff on the inside. What if we assume that the membrane prevents the stuff from moving, what will happen? But not water? So when we say that the membrane is semi-permeable, that means that the membrane prevents stuff from moving into or out of the cell, but it doesn't prevent water. I'm going to unhighlight that because, you know what? Don't be mean to my highlighter. Well, that highlighter doesn't. There we go. I'm just going to do this. There we go. Well, what can happen then is that instead of the stuff flowing, the actual water itself can flow in, and it can make that membrane swell out like that. And get those particles to spread out. So the particles naturally want to spread out themselves, and want obviously is anthropomorphizing what is a physical process, but the particles will tend to spread out. But if the particles can't spread out because they can't move across the membrane, what can happen is that water flows into the cell by osmosis to decrease the concentration of stuff inside the cell. Sorry? That's okay. So osmosis has the effect of diffusing the stuff inside of the semi-permeable membrane, but instead of doing that by letting the stuff out, which would let it spread out, it actually does it by drawing water in. Yeah. Yeah. Probably showing dialysis tubing. Yeah. We're going to use dialysis tubing later.
Something was filled with salt water and he was kind of like squishy and like stuck in.
You can do this tonight. If you want to see this in action with real cells, if you went home tonight and took a potato and sliced it into like sort of French fry shaped strips, don't cook them. You can cook some of them, but save 2 of them and you get 3 of them. You put 1 in pure distilled water, 1 of them in sort of just tap water, and then 1 of them in water with as much salt as you can possibly put in it. And the one that has a ton of salt, water is going to actually come out of the cells in the potato cells because they're surrounded by all that super salty water, and osmosis will want to spread out the salt in the water that you put the cell in, so it'll actually draw water out of the cell, and your potato will shrivel up. In perfectly pure distilled water, that'll be like this, where the water will flow in to try to spread out the stuff that's dissolved inside the cell, and your cells will actually, your potato piece will actually kind of like get really like swollen up with water. Yeah.
Why does it move outside of entropy
Yeah, so these particles, if there was no membrane there, they'd spread out, right? But the membrane prevents them from spreading out. So since the membrane prevents them from spreading out, the closest equivalent to those particles actually flowing out of the cell is that if water flows into the cell, that will cause the cell to expand. And if the cell expands, those particles spread out because the cell has gotten bigger. Now again, this isn't an active process. This happens naturally. So the cell isn't doing anything. This is a natural process. In the end, you could predict all of this by looking at individual particles bouncing into things and predicting what they're doing. Basically, the water will bounce into things, and it will have an easier time bouncing into things by moving into the cell than otherwise, because there's more like-minded water on the outside. Okay, so now this leaves the cell with a fundamental problem: controlling the movement of materials into and out of the cell. So, left to its, without some sort of barrier, the contents of the cell would simply diffuse away. With a non-permeable membrane barrier, materials couldn't get in or out of the cell. No. Non means not. Semi means sometimes, sometimes not. So we'll say a semi-permeable membrane allows some things to move in and out and others to not. Yeah.
I'll understand. So if you put the semi-permeable membrane and water without like the same material, nothing's gonna happen.
That's right.
Why?
I shouldn't say nothing will happen. Water will flow in and water will flow out. But if there's no difference between the water inside and the water outside, the rate of flowing out should be roughly equal to the rate of flowing in, and so you shouldn't really observe a noticeable change. But then if on one side of the membrane you add a bunch of stuff, now more water is going to flow towards the stuff than away from it by osmosis, and now it'll change the sort of where the water concentrates.
Would it also shrink it up?
Yep, exactly right. Yep, very good. All right. So, if you recall, we talked the other day about this structure. What's this thing? A phospholipid, which is, what is this spine section right here? I guess I should stop drawing there. These would have been H's before things bonded onto it. What's that molecule? the spine of a lipid molecule? Glycerol, very good. In a normal fat molecule, right, like animal fats and plant fats, all three of these will be attached to fatty acid chains. But in this case, two of them are attached to fatty acid chains, and the third one is attached to a phosphate group. And if we recall, the phosphate group is what? Highly hydrophilic, right? And this portion is highly hydrophobic. Okay, so we'll say we can say the head of this molecule will attract water while the tail repels it. And obviously referring to the head and the tail is sort of pejorative, but not pejorative, but rather vernacular, but it gets the idea across. In other words, if we had water and oil in a vessel, in a container, I guess oil usually floats. Typically oils are less dense than water, so we'll do that. So if we had water and oil in a vessel, and we were to put phospholipids in it, what would we find? We'd find that the phosphate heads would interface with the water and the tails would interface with the oil. Does that make sense? Now this is not what our cell is like, right? If we had oil and water, what's on the outside of most of our cells? Except for just the outermost layer, which your epidermis is interesting structurally. But typically speaking, most of your cells are surrounded by, sorry? That's what the surrounding is, but they're surrounded by water, right? We call it the intracellular fluid, or the extracellular fluid, rather. And then most of them contain what? They contain water. So that's good for oil and water, but how do you separate water from water, right? In other words, how would you get, if you had this whole thing with water, how would you get a separation there? What are the phospholipids going to do, right? Like, they can't line up like this, because if they line up like that, what side of the molecule is happy and what side of the molecule is unhappy? Sorry? Yeah, good. So if they go like this, why does that work? Kaylin's right. If we do that, this actually can work. We can separate water from water using this molecule. And why? Yep. Yeah, very good. So what will really be happening here then? Well, what we really are seeing here then is that that central section is nonpolar. And so you're not going to have water in there, but you can actually separate water from water. So a single layer would separate water from oil. But a double layer, also known as a bilayer, and if you look down below, you can see a diagram of this. Bless you. The interesting thing is, in water, will it actually form a straight line like that naturally? No. What would the favorable shape be? Well, we call it a micelle. So in water, which even just oil droplets will do this, right? If you drop oil into water, you'll find that it makes basically spherical drops in there until it rises to the top where it can collect in itself. Does that make sense? Why would it form a sphere if it's trying to sort of get away from water? Why not form a rectangle or a brick shape? Yeah. Good. If the oil molecules are repelled by water, they're naturally going to form a sphere because that has the least contact between hydrophobic oil molecules and hydrophilic water molecules. So naturally, in water, this layer will naturally form what we call a micelle, which is just a fancy way of saying a sphere. Sorry, what's that? So if there's water here and you start adding phospholipids to it, they will naturally form a spherical shape. And functionally what that means is we end up with an inner space. And of course, in our case, we would call that the cytoplasm, the cell cytoplasm. Which I guess is redundant since cytoplasm means cell fluid, but whatever, I'm gonna say it. Again, the important thing to understand is that you don't actually have to build the cell membrane into a sphere. You just have to build phospholipids and they will form a roughly spherical shape. And then you can do other things that will, adjust that shape, but naturally it will sort of form the sphere. Yeah, why not? Sorry? Yeah, so the phospholipid bilayer is the foundation of every cell membrane. Now, they can be made more or less permeable by adding other things. A cell wall, for example, will not completely take away permeability, but it'll make it less permeable. Things like that peptidoglycan layer that are on bacteria that we learned about will reduce the permeability of the cell. But the foundation of every cell is the phospholipid bilayer. That's what we call membrane. And then you can surround it with other things. Did that answer your question? That's right. This is the foundation of every cell's membrane. So when we say, so this is the structure of all membranes inside the cells. And that isn't just the cell membrane. What else? Where else does membrane occur in the cell? I shouldn't say inside. I'm just going to say in a cell because that actually includes the surrounding membrane. The nucleus, right? We have the nuclear membrane. So the nucleus itself is surrounded by membrane. What else? Yeah, the cell itself. What else? Yeah. Sure is made of cells, but let's think cell, not body here. That's not a wrong answer, like, because it's true, but in the end, that's just a bunch of cells. So individual, yep. Okay, we're gonna later. Okay, so we had a fire drill. We don't have time to talk about brain juice. So, sorry. All the organelles. So, like, the nucleus is just made of membrane. The chloroplast is just membrane. If you remember, does anyone remember the theory of how the organelles formed? Yeah, very good. And what's it called? Endosymbiosis. Very good. The idea that at one point a cell consumed another cell, but instead of it breaking down, it actually survived and thrived inside, and that's how you end up with the nucleus. And the same thing happens, and that's how you end up with a mitochondrion. So all of the organelles are also membrane bound. All right, so now we can say that so materials can move through the membrane more easily if they are what? Small is a huge advantage, right? Obviously, if you have to push your way through the membrane, which you can do. If you look at the structure of the membrane, it's what we call the fluid mosaic model. The idea that all of these phospholipids are just floating together, held together by forces. So something can shove its way through them and make its way through the membrane, but it's going to help if that thing is small. Large molecules will provide more hindrance, they'll have more trouble getting through. What else is helpful? The central portion of this membrane is polar and nonpolar. All these phospholipid tails, they are what? That's right, the nonpolar. So what kind of things will move through more easily? Nonpolar. And obviously that would include being uncharged, right? In fact, that should be a slash because it's really the same thing. And so if you look here, what are things that can move readily through the membrane? Small nonpolar molecules, such as oxygen gas, carbon dioxide, nitrogen gas. These things can flow right through the membrane. If you look here at the permeability, it's measured in 10 to the 0 centimeters per second. In other words, an oxygen molecule can move essentially through a centimeter of membrane, which is a ludicrous amount, a serious distance in just a second. On the other hand, something like water or glycerol, which are small, they're not charged, but they are polar. How well do they move through? 10 to the negative 4 means how much less permeable. Water, I guess, is about 10 to the negative 3, which is about 1,000 times less permeable. Does that make sense? We're using a logarithmic scale here. 10 to the 3 means 10 times 10 times 10 less permeable. So about 1,000 times less. glycerol about 100 times less again. And then about 1,000 times less again. Is glucose and sucrose. And finally, compared to the original, this would be measured in the hundreds of trillions times less permeable, which means one one chlorine ion will naturally move through the membrane in the same amount of time it takes about 100 trillion oxygen molecules to pass across. Does that make sense? In other words, functionally, these can basically flow right through. These can basically go right through. And can basically not go through at all. And we call this selective permeability. Now, to be clear, selective in this case shouldn't be understood to mean choice. Does that make sense? We're not saying that like the membrane sits there and chooses what can go through and what can't, but rather that the nature of the membrane is such that depending on the chemical properties of the molecule, they'll either pass through unobstructed or they will functionally not be able to pass through at all. Now, can the odd ion slip through a membrane? Yes. But functionally speaking, you're not going to get meaningful movement of ions across a membrane. How's that feel? Make sense? Oh, I was going to write that in that space there. So we don't need to write it again. No, there's not really anything I care to say there. So we're going to say each phospholipid in the membrane Each phospholipid in the membrane can move around and between the others and can be pushed aside by the other types. We call it the fluid mosaic model. So that is what makes it fluid. The fact that it's a mosaic, there is still always, they will maintain they maintain a barrier, however, because of the hydrophobic tails. I don't actually need that word fluid there. If you kind of space this out weirdly, you might want to highlight it and just move it. So in other words, these can freely move, they can switch around, but what they're not going to do is flip upside down or leave a big gaping hole, right? Because if they flipped upside down, the hydrophobic tails would be exposed to water, and the hydrophobic tails repel water. If they left a big gaping hole, the hydrophobic tails would be exposed to water, and they repel water. So even though they can flow freely, it maintains its structural integrity, and again, it does so naturally. It's self-repairing. So proteins, carbohydrates, lipids, and composites of all three can be embedded on the surface inside the membrane or all the way through the membrane. And this diagram shows that nicely. You can have some that sit just on top of the surface, some that travel all the way through it, some that sit embedded inside, and every combination thereof. You can see here, this is a carbohydrate. That would be a chain of what? Carbohydrate is a chain of sugars. What's a glycoprotein? It's a combination of protein, obviously, and, sorry? Not glycerol, though that's good thinking. Glycerol is actually so called because it's a derivative of, yeah, sugars. Glycogen is. So this would be an example of what we call a glycoprotein, where it's a protein that has embedded on top of it carbohydrates, right? So you can see the carbohydrate chain here of a pure carbohydrate. And then here we have what we call a glycoprotein, where it's a carbohydrate with protein on top of it. You can have what's called globular proteins. Protein channels, we'll talk about that. You can have things like cholesterol. Notice where cholesterol ends up. Cholesterol is lipid soluble, so it's found inside the actual inner layer of the membrane. You can have glycolipids. A glycolipid, the sugar portion will sit on top of the membrane, and the lipid portion of it will sit inside the membrane. Does that make sense? A protein can be on the inside or the outside because proteins are made of what molecule? What? Amino acids. And some amino acids are polar and some are nonpolar. All right. So we've got that diagram already. Proteins are embedded through the membrane and serve a variety of purposes. We have receptors that receive signals. We have machines that do work. We have enzymes. And we have a big one, transport. Remember on that list, what did it say can't move into or out of the cell naturally? Pretty much at all. Ions, for example, right? Like things with a charge. What moves very badly? Things like sugars and things like that, right? But do ions go into and out of our cells? Yeah, do sugars go into and out of our cells? Of course, we know they do, right? Like you can talk about your blood sugar levels because we can move sugars into your bloodstream or out of your bloodstream and into the cells they supply, right? So how do you move stuff into and out of the cell that can't get through the membrane? Well, you build a protein that will serve as a transport. Does that make sense? So proteins can be built by the cell that allow ions, large particles, etc., to move across the membrane. Yeah. Yep, that's right. Yep. So for the portion of the protein that's going to be embedded inside the membrane, the R groups on the outer surface of that would have to be the portion that's embedded inside the membrane. The R groups would have to be nonpolar. And then for the portion that's exposed to the outside and to the phosphates, you'd want that to be polar. Yeah. There are two main categories of transport proteins. Active transport, bind to materials and actively move them into or out of the cell. The fact that it's active means it requires energy. And we're going to say here, usually ATP, which if you talked about that in grade 10 science, great. If you didn't, don't worry about it. This is an energy molecule that the cell uses. And I'm just going to say we'll talk about it later. And this could be with diffusion. So this could be from low to high concentration. But it also could be from high to low concentration. It could go either way. Normally we said that which way does diffusion have to happen? High to low. But because it's active, because it requires energy, it actually can go from low to high. That's allowed. We do that all the time, right? If you take water and, put it in a bucket, you're putting it into a highly concentrated state. That can be done. So as long as you put energy in, you don't have to go with normal diffusion. This would be what we say against the concentration gradient. That's a term you're gonna hear a lot. The concentration gradient. This can go against the concentration gradient or with the concentration gradient. What's a gradient? Anyone know? Yeah, that's right. A gradient means that you won't have an area, here it's highly concentrated, here it's lowly concentrated. Usually it goes from high, and that's kind of high, and sort of high, a little high, it's medium, it's a little less than medium, it's pretty low, it's really low, it's very low, it's almost non-existent, and it kind of gradually moves from there to there. That's active transport. And these are proteins that are embedded there. In passive transport, these are usually just channels. Made of protein. And when I say a channel, what do we mean by that? Essentially, a hole. I'm sorry, it's concentration. I think I was thinking about the word gradient already. I get excited. I get ahead of myself. I'm sorry. They can still be specific. i.e. formed to allow some molecules and not others. Bless you. And that's a yawn, but still, I'll still bless you. Not that I have the power to bless anybody, but there we go. These are passive, which means they require no energy. And if there's no energy involved, which way do the things have to move? They have to go from high concentration to low, and do we call that against or with the concentration gradient? With. Very good. And again, does everyone remember what those square brackets mean? They mean concentration. Good. So active transport and passive transport, by the way, just diffusing across the membrane is also passive transport, but there can be a protein involved in passive transport and it can still be passive. If that protein just doesn't have to do anything that requires energy, if all it has to do is be a channel that is still maybe fitted to only allow certain things. So you have sodium channels in your cells that are sized so that sodium ions can pass through them, but nothing bigger gets through and nothing smaller gets through. The and nothing smaller gets through is interesting too, because as it passes through the top, it has to be the right size to cause the protein to sort of change shape, which will allow it to go through. So if it's too big, it just can't get in. But if it's too small, it actually doesn't trigger that sort of changing of shape that will allow it to pass through. So you can design, or the cell has evolved, channels that will only allow certain sized things and certain shaped things through that can still be passive, which is pretty cool. So you'll see these. Passive transport can just be diffusion. It can be just a channel. It can also be specific channels. And that's kind of what this is showing here, that this is sort of gated. Do you see that? How this channel here will only allow certain things to go through, whereas this one will just sort of let anything through. These are all still passive transport. On the other hand, active transport requires energy input. This is no energy input. And that's not completely true because there's some energy input, like, there's heat and other sources of energy, but there's no specific energy given to the protein to make things happen. Yeah. So in the case of the active transport, that energy is actually, oh gosh. The energy is actually used by the protein to mechanically move particles. Yes, sorry. So in active transport, it's not simply opening a channel and saying pass through. In active transport, it will bind to the molecule and then literally mechanically push it to where it's supposed to go. So you can think of a pump, or you can think of like a crane, something that is actively moving something from one spot to another. Does that answer your question? Cool. Sorry? So like anything else, a protein is built. So it would be built with one section that attracts this molecule called ATP. And then it would have to have something called a kinase functionality, which removes the phosphate group from it. You don't need to know that, don't worry about it. Long story short, it would have to be designed in such a way that when this molecule ATP comes near it, they're attracted to each other, they bind. And then it rips one of the phosphates off the ATP, which releases energy. And that has to be designed that when that happens, that energy gets absorbed by this molecule, and that causes all the rest of it to change shape and do a bunch of stuff. And that's the foundation of how proteins do things in the cell. Not just membrane proteins, but all proteins. They change shape and function. We'll talk about carbohydrates tomorrow. There's some follow-up questions here. You can answer those. Carbohydrates, do we have time? We don't have time. We'll finish that tomorrow. Well, probably not tomorrow. Tomorrow we're going to do our first lab, which means you need to have what done for tomorrow? Safety. Okay, then listen. The safety contracts are digital this year. Your family can sign them on online. But if you are tech-phobic or your parents are tech-phobic and you're worried about that, you can also come get a paper copy. But listen, I want your safety stuff done and your contracts signed by tomorrow. Okay? If they're not, will you be able to do the lab? No. All right, so if you want a paper copy of the contract, it's there.
Can I have something for that?
Yes. Yes, you can.
Diffusion, like, what is the morbidity?
Of which?
Morbidity, like rigor
Can you
mortis.
hold those, please?
Rigor mortis morbidity.
No.
It's like no, but rigor mortis.
Yeah, rigor mortis is basically when the muscle cells run out of energy after death. Oh, sorry, I thought I gave you one. Once your cells run out of energy after death, the muscles can't move anymore because both flexion and extension require the input of energy, and so.
Your blood wants to go where the least energy is, so it all just falls where your back is.
Right.